Yeah, so I thought I would take this opportunity to talk about some of the work in the lab, again, focused on high-resolution functional and physiological MRI, mostly in the human visual cortex. A lot of these technologies we think are really going to benefit, from the impulse scanner. And so, why are we all here today? I think that we're seeing, from many of the talks from earlier this morning, how much, EPI technology has improved over the past several decades. Again, EPI are a workhorse for fMRI and for diffusion as well. So these are some data from a recent review article. Um, so these are showing, the image quality in 2005. I'm not sure exactly how representative this is for EPI back then. You can see how much improvement we saw, in the past several decades, moving to both higher resolutions and higher image quality. But what we're seeing now, especially with the next gen scanners, is real step change in both imaging resolution and quality. And so the question becomes, with these such high imaging resolutions, are we really limited in our fMRI studies by imaging resolution or by the biological resolution of the technique? And so a lot of the work in my lab is really focused on seeing to whether we can understand a bit more about the relationship between hemodynamics and neural activity to try to extract more neural information from these high resolution data. And so with that, I'd like to talk a bit today about several topics. First, I'll talk about first principle biophysical models of fMRI responses. And there I'm going to ask, oh, excuse me, Alex. Oh, do you want to close that? Ah, cool. All right. Yeah, take your time. Awesome. Thanks for that. I appreciate that.
Is it loud enough?
Oh,
Louder? Can they hear?
Yeah. It's loud.
Okay.
folks online can hear okay?
Yeah, if you get a little closer, push the
Oh.
mic up.
Oh, and I will project. All right. Thanks so much, David. Thanks for the catch. Yeah, so I was saying that I'm going to start by talking about our first principle biophysical models of fMRI responses. And then we're going to ask, can realistic vascular models help us to link these unobservable microvascular dynamics to the observed fMRI signals? Then I'm going to talk about vessel-specific fMRI of blood oxygenation and blood velocity responses. And then we're going to ask, can fMRI measures in individual vessels teach us about hemodynamics and the relationship to neuronal activity? And at the end, I'm going to talk about quantifying both tissue and CSF displacement that correspond with the neural activity. And then we're going to ask, does neurovascular coupling play a role in neurofluid motion and brain clearance? And so just to start on biophysical models, now the biophysical modeling approach that we're using for this is so-called vascular anatomical network or VAN modeling approach. It really differs from conventional modeling approaches because it's based on realistic microvascular anatomy and dynamics. And so in this approach, we start with a single connected network of every capillary, and vein inside of one voxel here in the mouse sensory cortex, again, about 10,000 segments in this model. We can then ascribe boundary conditions, flow and pressure. The inputs to these models are single blood vessel dilations recorded from in vivo microscopy in response to neuronal activity. We then graft these onto the model, and we can use computational fluid dynamics to estimate the corresponding flow, pressure, and volume changes to neuronal activity. We can then calculate oxygen advection through the blood, oxygen exchange from the blood into the tissue, diffusion through the tissue in order to measure the blood oxygenation. We can then add that, to a bold kind of MR physics model to estimate the bold response to these dilations. So it kind of like this, it allows us to link, again, these unobservable microvascular dynamics to the observable fMRI signal. I think of this as, if we're sort of stealing some ideas from our colleagues in diffusion, we typically use these biophysical models to infer microstructure at scales smaller than the voxel size. Here we're using biophysical models to infer kind of subvoxel microcirculation in order to understand these fMRI signals. And so the first VAN modeling work actually provided a new insight into the bold response, this bold dependence on cortical orientation. And so this is work by Louis Gagnon and David Boas's lab. So although the orientation dependence of bold on a single blood vessel is well known, Louis, using his VAN model, quantified using simulations the orientation effect expected from a voxel containing realistic microvascular anatomy. In other words, he calculated how the bold signal amplitude varied with the orientation of the local cortical surface normal relative to the B0 axis. Now, while it might not be a surprise that some orientation effect exists, since the largest pupil veins run kind of parallel to the cortical surface, what he predicted was a sort of whopping 40% effect. That is, the bold response, should be 40% larger in cortical locations parallel to B0 compared to locations that are perpendicular to B0. This is such a large effect that he immediately went down to a conventional 3D scanner, looked at whole brain bold activation in response to a CO2 stimulation, and saw a similar effect in these conventional, very ordinary bold fMRI data as well. So I think this is quite cool because it shows us that this van modeling approach can make a new prediction about the bold signal based on these realistic van models derived from mice. And so the idea here is that, can we derive similar van models for the human cortex to gain similar insights into human fMRI? And so while, the trouble with these van models is that they require both accurate microvascular anatomy and dynamics. And while it's possible to reconstruct the microvasculature using in vivo microscopy techniques in mice, a little bit harder to reconstruct these in humans. So we've taken a different approach in which we synthesize these van models of human vasculature based on anatomical distributions taken from the literature. So just to show you an example of the scale, on the left, I'm showing a current mouse van, and on the right, the human van that we're using for our fMRI simulations. And so with these human van models, we can start to understand how differences in mouse versus human vascular architecture might impact the bold timing and different features of the hemodynamic response. We know that the vascular architecture differs a lot between mice and humans, both in terms of their topologies. We know that in mice, the artery to vein ratio is about 1:3. In humans, it's about 3:1, as well as their geometry. In humans, we have much larger vessels with much longer lengths. So we took our mouse van models and our human van models, we simulated the bold responses using each. We saw was that, compared between the rodent and human responses, the human responses were much slower than the rodent responses. And at first, we weren't sure why this was the case. We then went back to the literature and saw that, in fact, people do measure bold responses in humans that are much slower than those in mice. And it looks like the reason for this slowness is mostly because of the longer lengths of the blood vessels. It just takes longer for the upstream propagation of dilation to reach the cortical surface, originating in the parenchyma. We think that this, longer time for the dilation is causing the slower bold response in humans. So I think this is quite cool. It's showing us that these van models allow us to investigate how different features of the functional vascular architecture may impact measurable features of the bold timing. Now, at first, when we started to generate these synthetic bold van models, we thought this was kind of a workaround, that allowed us to get around the difficulties in reconstructing van models from microscopy data. I actually found that this synthetic approach could actually be quite useful because it allows us to parametrically vary individual features of the vascular architecture and see how these features may impact the bold responses. So in this case, we started off with a van that had high capillary density in layer 4. You can see this in the rendering on the top, as well as this histogram in the middle. If we simulated the bold response using this van model, we saw that we look at the peak of the bold amplitude as a function of cortical depth. We see that there's a strong peak near to the PO surface. But we also saw the sort of local peak of the bold response in the location of the high capillary density in the middle layers of the cortex. But now we can start playing games. We can start synthesizing van models that have high capillary density at the top half of the cortex, a van model that has high capillary density in the middle third of the cortex, a van model that has high capillary density in the lower half of cortex. And in each case, we see that there's a local bump of the bold response at the location of the high capillary density. And this is quite cool because we know a lot about these macrovascular biases in bold fMRI. We see stronger bold responses in locations where there's more large PO veins. However, this is indicating to us that there may be microvascular biases as well. And so this is kind of difficult to interpret because, as we know, it's quite common that vascular architecture reflects functional architecture. We know, for example, that we tend to see high capillary density in locations where there's high baseline neuronal activity and a high baseline metabolism. And so oftentimes the vascular architecture could reflect the functional architecture. So it's a little bit hard to interpret these bold responses given the fact that there's this relationship. But I think what's cool about these models is that they allow us to sort of quantify how much of these cortical depth profile effects we're seeing that are attributable to the vascular architecture to help us to understand how much of these effects can be attributable to the neural activity itself. So we think this is quite cool. Okay, so next I'd like to talk about vessel-specific fMRI and blood oxygenation and velocity responses. And so this vessel-specific, or also known as single vessel fMRI, is introduced by my former MGH colleague, Jinyu, for both mouse, bold, and CBV fMRI. So the idea here is that we take a single MRI slice with high in plane resolution, positioned within and tangential to the cortex, that allows us to look at voxels at the scale of individual vessels, as shown here. This allows us to measure hemodynamics from individual arteries or veins. And so again, with the conventional voxels, we tend to see that all voxels contains all vessel types, arteries, capillaries, and veins. But with this vessel-specific approach, we use these anisotropic voxels to retain SNR. They're perpendicular to the vessels to measure their cross-section. It allows us to measure roughly one mesoscale vessel per voxel. And so why do we want this vessel-specific fMRI in humans? Well, it provides us with quantitative measures of vascular dynamics, which provides direct validation with other modalities such as microscopy, going from kind of MR-specific measures of BOLD, ASL, and vaso, to meaningful physiological parameters such as SO2, velocity, and vessel diameter. I think this provides improved interpretability, helps us to understand the fMRI signals and study neurovascular coupling directly in humans. I think they're also providing us with more appropriate inputs to and validations for our human band models. And so just as a warm-up here, we wanted to see whether we could use vessel-specific fMRI to look at blood oxygenation and velocity responses in arteries. We know that BOLD fMRI mostly measures deoxyhemoglobin changes, which are concentrated in veins, but we think that arteries might also contribute to BOLD fMRI. That's because at baseline, arterial blood is not really fully oxygenated. So any changes in blood velocity are going to cause changes in the BOLD signal within arteries. We also know that a lot of oxygen actually exchanges from the blood into the tissue around these large arteries. It doesn't only exchange with the capillary bed. So changes in metabolism could also cause changes in BOLD signal around arteries. So I wanted to see with these vessel-specific approaches whether we could detect arterial BOLD. And so with this approach, we started off with an anatomical scan, kind of high in-plane resolution, 0.25. The fMRI data here were 0.4 millimeter in-plane multi-echo flash. So this is a flash acquisition, just a single slice. So it has poor temporal resolution, about 4 seconds, but we can present the stimulus with a little bit of jitter to be able to estimate or sample the hemodynamic response at a grid about 0.8 seconds. I just looked at flashing checkerboard activation. So here on the top row, I'm showing you how we're able to identify individual vessels. It's single slice. So with a short echo time, we're getting a lot of inflow effects. All the vessels are bright. At longer echo times, the veins darken. And so we can use this to identify individual arteries and veins. On the bottom row, I'm showing you the overlaid fMRI data, again, showing you how we can identify fMRI signals within individual vessels. And so here some of the responses are both inflow-weighted and BOLD-dominated responses. Again, the short echo times give us more kind of inflow-weighted fMRI responses. The long echo times give us BOLD-dominated. We can look at both short-duration stimuli and long-duration stimuli in individual arteries and veins. I think this is already pretty cool, right? Because it's showing us that, first of all, there is BOLD in arteries. I think that's, very helpful to see. We're also seeing that there is a lot of BOLD in veins. I think people often assume that at 7 Tesla, because of the short T2 stars of the intravascular signal, we shouldn't expect to see any intravascular BOLD. We're seeing that we can identify this quite clearly in these data. So we can normalize these to look at the timing between the arterial responses and venous responses. It's quite cool. What we're seeing is that the responses to the short duration stimuli appear to exhibit kind of very similar timing between arteries and veins. It kind of makes sense. We know that for longer duration stimuli, pressure kind of builds on the venous compartment. We see a compliant kind of ballooning effect and a local decoupling of the arterial and venous signals. Whereas when we go to shorter stimulus durations, the balloon doesn't have the time to engage. So I think this is quite cool. We've seen this a lot in optical imaging. Makes sense to see this in the fMRI data. It shows us that the hemodynamic responses in veins can better reflect the responses in arteries when we use short duration stimuli. I think this is a nice insight, giving us some ideas about experimental design, helping the venous BOLD signal more reflect the arterial signal, which is again the site of neurovascular coupling. We think this can provide us with increased neuronal specificity. Again, using veins as a readout for arterial dynamics. We've also been using other vessel-specific fMRI approaches, including looking at blood velocity responses and supplying arteries. This is kind of an off-the-shelf phase contrast angiography technique that we've optimized to perform fMRI. So in this case, we're using a 0.8 millimeter in-plane single slice temporal resolution of 2 seconds with a Venk of 60 centimeters per second. We position this able to looking at responses within the posterior cerebral artery, which supplies visual cortex, looking at velocity responses to flashing checkerboard stimulation. We saw in individual vessels that the blood velocity in these upstream arteries increases from about 28 centimeters per second to about 32 centimeters per second, or about a 20% velocity increase. So when I first saw this, I was quite puzzled. So if you note on the top left corner, this slice that I'm showing you is about 6 centimeters away from the activated cortex, right? And we're seeing this, hemodynamic response very far away from the site of neural activity. I think that, maybe some of this is just mass balance. If there's increased perfusion into the tissue, the blood has to come from somewhere. So it kind of makes sense that there would be increased flow in these upstream vessels. We also think of this as a form of a network level effect. The hemodynamics are really coupled quite a lot across different brain regions, more than it really is appreciated. There's a lot of interdependence of hemodynamics across the brain. And so we think that tools like this can give us some insights into neurovascular coupling at larger spatial scales. Okay, so I'm going to conclude by just showing some work on physiological MRI quantifying tissue and CSF displacement. And so the starting point here is some recent work from Laura Lewis looking at coupled low-frequency hemodynamic oscillations in CSF flow. And so what Laura did here was she acquired kind of a simple kind of bold acquisition covering the cerebrum. And what you're able to see is these changes in inflow signal at the fourth ventricle, shown here by the red arrow. And she's able to look at concurrently kind of CSF dynamics as well as hemodynamics. What she saw, especially when subjects were sleeping, she saw this kind of interesting kind of anti-correlation between the kind of bold oscillations as well as the inflow oscillations at the fourth ventricle. Again, CSF flow changes. Makes sense. Again, bold is related to changes in blood volume. As the blood volume expands, it kind of pushes the CSF out of the brain. As the blood volume kind of constricts, it kind of pulls the CSF back up. And so she came up with a very kind of simple model to link these EEG signals that she also measured concurrently with the fMRI, these kind of neuronal changes that feed into changes in CBF, which cause changes in both blood volume and bold, and then changes in CSF. So we want to understand this pathway a bit more closely and ask whether cortical displacement actually may mediate the CSF motion driven by changes in blood volume. So the idea here is that with neuronal stimulation, we might expect that blood volume is going to increase, which is going to cause a cortical expansion and some change in CSF flow. And so we wanted to test this out. David, I'm showing this partly because you kind of pioneered a lot of this tissue and CSF displacement imaging. There's many both classic and modern MRI methods for measuring tissue displacement. We're going to use the DENSE technique. This is used for measuring tissue strain, but here we're just going to apply it to looking at tissue displacement. It's a really simple sequence. It's basically a steam diffusion sequence looking at the phase. It's a stimulated echo, phase-based motion encoding for looking at tissue motion quantification. Again, the DENSE method, primarily used for measuring tissue displacement, but here we're going to use it to look at displacement in response to neuronal activity. So the motion encoding mechanism used here is really analogous to what we use in phase contrast angiography. What's cool about DENSE, though, is that we can achieve a really small minimum VENC, less than about 1 millimeter per second, compared to phase contrast, which can only achieve a VENC of about 4 centimeters per second. So this allows us to get sensitivity to really slow velocities, really subtle displacements. What's cool about this technique, though, is that, if we look at the magnitude of the B equals 0 images, they're basically stimulated echo bold. So just to show you this, first on the left, I'm showing you some kind of conventional gradient echo bold in response to visual stimulation. And on the right, in the same subjects, I'm showing you this stimulated echo bold measured, again, using the magnitude of these DENSE data. So stimulated echo bold is not really looked at very much. It's kind of similar to spin echo bold, has more specificity, lower sensitivity. So you can see here that it took us, 45 trials to measure the bold with stimulated echo compared to just, 5 trials of the gradient echo bold. But it shows us that we can measure these hemodynamics directly using the magnitude of the DENSE. But what's cool is that the phase of the DENSE provides us with a quantitative measure of tissue velocity and displacement. As we think about, first of all, what might be the expected patterns of cortical tissue motion, we might expect really that the cortical tissue is really going to move the most during the transience of the hemodynamic response. So the expected temporal pattern, we'd expect to see tissue velocity should be highest when the blood volume is changing. So the pattern should look like the temporal derivative of the bold response. And indeed, it does look like this. We see greatest tissue motion in the visual cortex in response to visual stimulation at stimulus onset and offset across many different ROIs tested. We quantified this tissue motion corresponds to about 30 microns of displacement, again, stimulus onset and offset, which is similar to what has been measured previously. But again, what I think is cool about this technique is that we can concurrently measure both hemodynamics and tissue displacement. Again, if we look at the magnitude of dents, we can kind of measure the hemodynamics. We can take the temporal derivative of the hemodynamic response. This gives us a sense for what the tissue displacement should look like. And then we can look at the phase of these same data and quantify tissue displacement, and we see that they actually align pretty closely. So this is really helpful. It's one thing to be measuring, tissue responses to visual stimulation, where there's a trial-locked response. But if we want to look at kind of spontaneous or ongoing oscillations, we need to be able to measure these hemodynamics and tissue displacement at the same time to understand how they're linked. Okay, so just to recap, I've shown you how biophysical models can provide us with insights into microvascular influences on fMRI signals. These differences in vascular architecture we see between mice and humans, do give rise to differences in hemodynamics between these two species. And shows us that the mouse is still the most common model used for studying neurovascular coupling or understanding neurovascular coupling in humans. I think that there could be a lot of differences due to these architectural differences between the species. And so I think there's a need for human measurements to understand neurovascular coupling in humans. I've shown you how vessel-specific fMRI provides direct measures of hemodynamics within individual arteries and veins. It demonstrates how arterial bulge responses can be observed in small PL vessels, and ongoing work is integrating these direct hemodynamic measurements provided by vessel-specific fMRI in humans as inputs to and validations of our animal models. I showed you how dense fMRI provides concurrent measures of tissue displacement and bulge during neural activation, which again suggests that neurovascular coupling contributes some of the mechanical energy that's needed to drive kind of CSF, ISF, neurofluid motion across the brain. And we're now working on separating out these tissue and CSF movements with smaller voxels. And just to wrap up, yeah, so I think this mesoscale fMRI, it's not just high resolution. I think that this mesoscale fMRI really provides a bridge between scales, a bridge between modalities. It's quite exciting to be able to use this to better interpret our fMRI data, and again, to understand the physiological processes that occur at scales below those of conventional muscle sizes. And so with that, I will stop. So thank my lab for all of their help in contributing slides, collaborators, our funding sources for their generous support, and thank you guys for your attention.